So the second session was about biomarkers, and there were presentations. You probably all remember from Frank Sharmilla in Virginia, academics who talked about both imaging and biochemical markers. Frank Romer talked about X-ray versus MRI in large part, showing that X-ray gave you a heterogeneous and not very accurate sometimes picture of what the structure of the joint was, and that MRI morphologically presented more accurate information. Then Sharmilla went into data that evaluated imaging biomarkers as predictors of all kinds of outcomes, of cartilage loss outcomes, of pain outcomes, of knee replacement outcomes, and showing variable performance, but promising performance especially for machine learning type outcomes in various data sets. And then Virginia did essentially something similar for biochemical markers, reviewing both the FNIH studies and other studies looking at systemic or blood and urine biochemical markers and their relation to either cross-sectional or longitudinal outcomes that are either imaging or clinical, and showed variable results, both remarkably detailed, comprehensive discussions in a short period of time of lots of data, suggesting there was promise for any number of both imaging and biochemical markers. I think with respect to biochemical markers, it was urine CTX2 and this new marker, whose acronym I won't remember, but where there needs to be more data and and there is some promise. And then there was a whole other series of presentations, starting with Nikolay, and then who laid out a larger number of types of biomarkers than I had thought about before and what they were. And then Jeff Siegel and later Thomas Fleming and Greg Levin, who sort of helped us understand what how the FDA interprets proof for and qualifies various biomarkers. The surrogate biomarkers being the ones obviously of greatest interest, but the ones where the bar is really high for us, where there need to be multiple trials where those biomarkers were measured both at the beginning and after treatment to see that the biomarkers changed in concert with treatment responses and did so consistently. And an example of blood pressure and stroke or blood pressure and cardiovascular outcomes was shown, and other examples were shown too, some where the biomarker frankly didn't predict consistently the therapeutic effects of the known therapeutic effects of an agent or of comparative agents. And so I think it's fair to say that we were left with the notion that we aren't anywhere near using surrogate biomarkers in osteoarthritis. And then Tom Fleming and Greg Levin, and I think to some extent Nikolay and Jeff, both differentiated prognostic versus predictive biomarkers, which was something I hadn't really thought about much personally, which it sounded like we're really in the realm of prognostic biomarkers, which Tom Fleming called low-hanging fruit, which I thought was sort of appropriate probably, but where there were correlational analyses looking at the relations at baseline of our biomarkers with outcomes that were collected over time. That we didn't necessarily even have predictive biomarkers. So why don't I stop there? Nikolay, let me defer to you because I know I suspect you may want to say more about biomarker qualification from the FDA's perspective.
Thanks, David. I think it was an outstanding summary of the session and the discussion. I don't think I have a whole lot more to add, other than just to emphasize that this is more of a realistic review of the landscape of biomarkers in osteoarthritis. But we as an agency are certainly open to work with all stakeholders to advance this and the knowledge and the understanding of these biomarkers so that they can be used in more than just predictive and prognostic context. I think this summarizes our session, and I think it's very tightly intertwined with the discussion in the last session, which is how to assess long-term benefit and how to potentially provide the data needed to support something as a surrogate endpoint.